Today we're going to be talking about the oxygen dissociation curve. The oxygen dissociation curve is a graph that we look at when we're thinking about how haemoglobin, which is found in red blood cells, carries oxygen around the body. The graph has a sigmoid shape, which we call a sigmoid curve, which overall looks like an S shape, which you can see here. The x-axis shows the PO2, which stands for the partial pressure of oxygen. The partial pressure of oxygen is the concentration of oxygen within a mixture of gases. You can think of it as the concentration of oxygen overall. The y-axis shows the oxygen percentage saturation, or you can think of it as haemoglobin's affinity for oxygen. So in my previous video on haemoglobin, I talked about its cooperative binding, which is when one oxygen molecule binds to haemoglobin, it changes the shape of haemoglobin, making it easier for subsequent oxygen molecules to bind. So if you haven't watched that video already, please go back and watch the video called The Structure of Haemoglobin. So therefore it increases haemoglobin's affinity to bind to oxygen, or you can think of it as increasing its attraction for oxygen. So you can see here, at low levels of partial pressure of oxygen, the haemoglobin has a low affinity for oxygen. As the partial pressure of oxygen increases, more haemoglobin becomes bound to the oxygen because of its cooperative binding ability. This makes it easier for more oxygen molecules to bind to the haemoglobin, and you can see a massive increase in the slope of the graph, showing an exponential increase in oxygen saturation. This last part of the graph shows at very high levels of the partial pressure of oxygen, the haemoglobin has become saturated with oxygen. So this shows 100% saturation because haemoglobin is filled with the oxygen molecules, forming oxyhemoglobin. Oxyhemoglobin. Now we're going to talk about the Bohr effect. It's B-O-H-R, the Bohr effect. It's named after a scientist who discovered that in high carbon dioxide levels, or high carbon dioxide conditions, or low pH levels, which would be acidic environment, it causes haemoglobin to release oxygen more readily. And this causes the graph to shift to the right. So this would happen, for example, in the tissues where cellular respiration is constantly taking place, releasing carbon dioxide as a waste product and therefore increasing its concentration. So on the graph, it would be here, this dotted line, I'm just going to colour it in red. This would be our right shift, and it's most likely to take place in the tissues of the human body. So let's look at the Bohr effect in more detail. The goal in the tissues is to get as much oxygen off the haemoglobin as possible for cellular respiration to make ATP. Remember that ATP is the energy currency found within the cells. So if you remember the word equation for ATP, if we wanted to do the chemical formula for that, it's C6H12O6 plus oxygen produces carbon dioxide plus water plus energy in the form of ATP. So if we wanted to balance that equation, it would be 6 oxygens and 6 carbon dioxides produced, as well as 6 water molecules. And overall, we've just got ATP here, but this reaction usually produces around 36 to 38 molecules of ATP per molecule of glucose during aerobic respiration, as long as oxygen is present. The carbon dioxide is going to diffuse out of the tissues and into the plasma of the bloodstream. Carbon dioxide in the bloodstream combines with water to produce carbonic acid, and this reaction is catalyzed by an enzyme called carbonic anhydrase. Carbonic acid is just an intermediate, and it quickly dissociates to form hydrogen ions and bicarbonate ions. Let's draw that equation. So we have carbon dioxide plus water produces carbonic acid. Carbonic acid, H2CO3, which then quickly dissociates to form hydrogen ions and bicarbonate ions. This reaction is reversible, which you can see from the double arrows here, and that is important for maintaining pH balance in the blood. In general, hydrogen ions in solution lower the pH of any solution, so hydrogen ions in the blood are going to lower the pH of the blood. The hydrogen ions also can bind to the haemoglobin, which causes a conformational shape change. Anything that binds to a protein molecule usually causes some shape change, and that's going to allow the haemoglobin molecule to release oxygen. And so overall, this is what we term the Bohr effect. We've also got a molecule called 2,3-bisphosphoglycerate, or it gets called BPG for short. You might also hear it get referred to as 2,3-diphosphoglycerate. We call it 2,3-BPG for short. It's an intermediate in glycolysis, and it's produced in excess in conditions of low oxygen concentration, such as high altitude, anemia, or if someone's exercising. It binds to haemoglobin and red blood cells, which reduces haemoglobin's affinity for oxygen, again making it easier for haemoglobin to release oxygen to the tissues. So overall, we can see if we've got a higher environment of carbon dioxide, or we've got a lower pH and acidic environment, or if there's a higher temperature or increased levels of 2,3-BPG, then we're going to have a right shift in the oxygen dissociation curve graph. This high temperature is a result of cellular respiration and metabolism taking place in the tissues. Now that we've looked at what happens during a right shift in the oxygen dissociation curve, let's look at what happens during a left shift in the oxygen dissociation curve. A left shift occurs when haemoglobin develops a higher affinity for oxygen, meaning it holds onto oxygen more tightly. As the partial pressure of oxygen increases, haemoglobin continues to bind to oxygen due to its cooperative binding ability, but because of its high affinity, haemoglobin doesn't let go of the oxygen as easily. The slope of the graph increases steadily, but it takes longer for the oxygen to be released into the tissues. Let's just draw this left shift in here over the dotted line so you can see it more clearly. There we go. And then up at the top here, at very high partial pressure of oxygen levels, haemoglobin becomes fully saturated with oxygen, reaching 100% saturation, where it is completely loaded with oxygen molecules, again forming this oxyhemoglobin. A left shift occurs in the oxygen dissociation graph under conditions where haemoglobin's affinity for oxygen increases, and this can happen when we've got lower levels of carbon dioxide, or we've got a high pH, so alkaline levels, or somebody suffering from alkalosis, or if we've got lower temperatures, but also when we've got low levels of the 2,3-BPG. These kind of conditions would take place inside the lungs. For example, in the lungs where oxygen levels are high, carbon dioxide levels are low, and the pH is slightly alkaline there. The left shift helps haemoglobin hold on to oxygen more tightly so it can transport oxygen efficiently throughout the bloodstream. The lungs have a lower temperature compared to the tissues because they're exposed to cooler inhaled air. They also have a lower metabolic activity, and blood circulation also helps dissipate the heat, given that overall lower temperature compared to the tissues. This cooler environment contributes to the left shift in the oxygen dissociation curve, increasing haemoglobin's affinity for oxygen. This adaptation helps haemoglobin hold on to oxygen tightly in the lungs for efficient transport. Meanwhile, like we saw before, in the warmer tissues where heat is being generated by cellular respiration and metabolism, oxygen is released to meet the body's energy demands. So that wraps up the oxygen dissociation curve, showing how haemoglobin adjusts to efficiently carry oxygen from the lungs to the tissues. Thanks for watching, and I hope this explanation was helpful.